All right, let's go. Number 10. The Hadza gut gap and what it quietly erased. The mystery is not what killed the ancient world. It is what the ancient world was quietly keeping alive inside us, and what happens now that it is gone. In 2016, Justin Sonnenberg and his team at Stanford University compared the gut microbiomes of the Hadza hunter-gatherers of Tanzania with those of Italian urbanites living ordinary modern lives. The contrast was not subtle. The Hadza samples contained bacteria from genera entirely absent in the Western adults tested, including treponema, organisms long associated with breaking down the fibrous wild tubers that Hadza foragers dig from the cracked red earth of the Rift Valley. Over 600 distinct bacterial species were identified in the Hadza samples alone. That number is staggering. Modern Western microbiomes carry nothing close. 2 years earlier, researcher Jeff Leach of the Human Food Project had spent extended time embedded in Hadza territory in Tanzania, documenting the gap from another angle: dietary fiber. Ancestral humans likely consumed between 100 and 150 grams of dietary fiber per day, drawn from roots, seeds, bark, and foraged plant matter, compared with an average modern Western intake of roughly 15 grams. The gut bacteria responsible for fermenting that fiber, including Ruminococcus champanolensis and related species, have become functionally extinct in many modern Western populations, not through illness or infection, but through simple disuse across generations of industrial eating. Then, also in 2016, Ron Sender and colleagues at the Weizmann Institute of Science in Rehovot, Israel, published a revised census of the human body. Their finding placed the total number of microbial cells in the human gut at roughly 38 trillion, approximately matching the total number of human cells in the body. The human being, it turned out, was never purely human. It was always a colony. But here's where the standard explanation falls apart. The conventional framing treats the loss of microbial diversity as a side effect of progress, a minor casualty of clean water, refrigeration, and antibiotics, net positive and largely irrelevant. What Sonnenberg's comparison and Leach's fiber data together suggest is something more structural. Modern Western microbiomes have lost an estimated 1/3 of the ancestral microbial diversity documented in hunter-gatherer populations. 1/3 of an inheritance shaped across millions of years of human evolution vanished within a handful of generations, without ceremony, without notice. Skeptics argue that higher microbial diversity is not automatically healthier. Some Hadza-exclusive bacteria may simply reflect local diet, not ancestral advantage. That caveat deserves honest weight, but the functional disappearance of entire genera, not merely reduced counts, points to something beyond dietary preference. This means the gut arriving in a modern body is not a degraded version of the ancestral gut. In measurable terms, it is a different organ. The immune signaling, the metabolic chemistry, the fermentation capacity, all of it calibrated by evolution to a fiber load and a microbial landscape that most modern humans will never encounter. This connects directly to the through line of the ancestral toolkit. The survival techniques that kept early humans alive for hundreds of thousands of years. The foraging, the tuber digging, the consumption of raw, fibrous plant matter were not merely cultural practices. They were the maintenance schedule for an internal ecosystem. Removing the inputs collapsed the system. That ecosystem, in many modern guts, no longer exists in a form capable of running the original software. What makes this particularly haunting is that the loss was never a decision. No individual chose to surrender 38 trillion years of accumulated microbial partnership. It happened incrementally, invisibly, across generations that had no framework for understanding what they were relinquishing. The Hadza territory is still there, the red soil still yielding its fibrous tubers to people who dig with wooden tools. The 38 trillion cells are still counted. The census still stands. What remains unresolved is the quieter question beneath all the data: what exactly was lost when the organisms departed, and whether the body still carries some faint record of the world they were built to inhabit. Number 9. The mammoth-fat paradox, and the mystery runs deeper than anyone expected. In 2008, archaeologist Jiří Svoboda of the Academy of Sciences of the Czech Republic was conducting isotopic analysis at Dolní Věstonice, a 26,000-year-old occupational site in Moravia, when the bone chemistry stopped making sense. The site had already yielded extraordinary finds, ceramic figurines, elaborate burials, the dense refuse of long habitation. But the dietary signal in the bones was the thing that demanded explanation. The isotopic record told a clear story. The hunters who camped at Dolní Věstonice were eating woolly mammoth fat in substantial quantities. Mammoth fat runs to approximately 70% saturated fatty acids. That single figure demands a pause. 70%. A modern cardiologist shown that dietary profile without context would predict severe irreversible atherosclerosis in the population within 2 generations. The bones at Dolní Věstonice showed none of it. No calcification consistent with metabolic syndrome. No skeletal signatures of the arterial damage that such a diet fed to a modern Western body would almost certainly produce. The textbook picture before this analysis was straightforward. Prehistoric humans survived on high-fat diets because their lives were short and violent. They simply died before chronic disease could take hold. The fatty acids were the problem. Early death was the solution. It was a grim but tidy arrangement that most researchers had quietly accepted. But here's where the standard explanation falls apart. The bones at Dolní Věstonice are not the bones of people who died young from other causes before metabolic damage could accumulate. The skeletal population includes individuals who lived well into middle age. The damage is not merely absent in the young, it is absent across the board. This means the body processing that fat was not the same body that would process it today. Svoboda's isotopic findings, read alongside later microbiome research, pointed toward a biological negotiation happening below the level of anatomy. Maria Gloria Dominguez Bello and her team at New York University documented in 2015 that the Yanomami people of the Venezuelan Amazon, among the most isolated human communities ever studied, carry gut microbiomes roughly twice as diverse as those of average adults in the United States. More striking still, some researchers argue those Yanomami microbiomes contain functional genes capable of degrading modern agricultural pesticides, despite the Yanomami having had zero pesticide exposure, suggesting the ancestral microbiome held metabolic capacities that industrial life has quietly erased. Mainstream researchers note this finding is compelling but not yet fully explained, and the pesticide gene interpretation remains under active debate. What the evidence does support, with increasing confidence, is that a diverse ancestral microbiome could generate short-chain fatty acids in quantities that modern Western guts simply cannot match. Those short-chain fatty acids modulate inflammation, regulate lipid metabolism, and appear to blunt the cardiovascular damage that saturated fat would otherwise cause. The Dolní Věstonice hunters were not immune to fat. They were equipped to process it. The napping floors at sites like Talheim in Baden-Württemberg, Germany, where Christian Meyer of the Senckenberg Center for Human Evolution documented pulmonary calcification in Neolithic farmers through skeletal CT scans in 2015, add another dimension. Flint napping releases silica dust particles below 10 micrometers fine enough to lodge permanently in lung tissue and cause silicosis. By modern occupational safety standards, a Paleolithic tool-making workshop would be classified as immediately dangerous to life. The Talheim individuals show the scarring. They worked in those conditions and survived them long enough for the calcification to become visible in bone. The body they carried negotiated terms we can no longer offer. What makes this particularly haunting is that the negotiation did not end with the Paleolithic. It is ending now in real time as microbial diversity continues to decline across industrialized populations with each successive generation. The bones at Dolní Věstonice are still held in Czech collections, still measured, still isotopically sampled. The fat content of a mammoth haunch is still calculable. What remains unresolved is the quieter implication that the body capable of surviving that diet without consequence is not a relic of the deep past, but something the living world has been slowly and perhaps irreversibly dismantling. Number 8. The Neanderthal mouth and the mystery it left behind. In 2017, Laura Weyrich and her team at the University of Adelaide were analyzing dental calculus scraped from Neanderthal teeth recovered at Qesem Cave, a limestone network buried in the green hills of Asturias in northern Spain. The samples were roughly 48,000 years old. What she expected was a biological record of diet. What she found was something considerably stranger. The calculus, hardened plaque, essentially fossilized bacteria, had preserved ancient DNA in extraordinary condition. Qesem is cold and stable, and that stability had protected the microbial record with unusual fidelity. Inside the samples, Weyrich identified genetic signatures of Methanobrevibacter oralis and related archaic archaea, microorganisms so rarely detected in modern human mouths that when researchers encountered them in these concentrations, they had to construct new classificatory language just to describe what they were looking at. The Neanderthal oral cavity was not simply an older version of the modern human mouth. It was a different environment entirely populated by microbial communities that now exist, if at all, only at the margins of the human body. That single fact deserves a moment of stillness. Before this analysis, the working assumption was broadly this: Neanderthal biology differed from modern human biology in anatomy and perhaps in cognition, but the basic architecture of digestion was assumed to be roughly continuous. The mouth was a mouth. The gut was a gut. The microbial passengers were considered incidental detail, not a load-bearing part of the physiological story. But here's where the standard explanation falls apart. If the oral microbiome of a Neanderthal was not simply ancestral to ours, but was genuinely distinct, if those archaea functioned as active pre-processing agents, priming food chemically before it ever reached the stomach, then digestion itself was a different operation in a different body. The mouth was not just an entry point. It was the first chamber of a system calibrated over hundreds of thousands of years to handle food that modern dietary science would describe, in clinical understatement, as challenging. Consider what that system was processing. Anthropological and zooarchaeological work documents that prehistoric foragers consumed raw animal tissue, fermented plant matter, and in many documented contexts, materials that would register as dangerous by any contemporary measure. Research by Sarah Young at Cornell University, published in 2011 and drawing on evidence from Neolithic sites in the Limpopo Valley in southern Africa, documented systematic geophagy, the deliberate ingestion of soil and termite-excavated earth, among prehistoric foragers. That soil carried quartz silica particles, microbial spores, and helminth larvae simultaneously. The body absorbed calcium and iron from those loads. This is controversial territory, and mainstream archaeologists continue to debate the degree to which this was dietary strategy rather than incidental contact. But the physical evidence at Limpopo is documented and material. The important point is what Young's analysis implied. A gut primed by a radically different microbial starting environment may have processed those loads as nutrition. The same loads, introduced into a modern dysbiotic gut, would represent a serious and potentially fatal toxic event. The mechanism for that dysbiosis is not hypothetical. Research led by Eran Segal at the Weizmann Institute of Science in Rehovot, Israel, published in Nature in 2018, measured gut microbial diversity in individuals undergoing standard antibiotic courses and found reductions of up to 30% within 72 hours, with some key anaerobic species failing to recover even after 6 months of normal eating. A single clinical intervention, one course of medication, and the ancestral architecture is measurably and sometimes permanently diminished. This means the dismantling was not gradual or abstract. It was chemical, it was recent, and it was deliberate. What makes this particularly haunting is the sequence it implies. A pre-processing oral environment calibrated across deep time primed a gut capable of surviving exposures that would now be lethal, and modern medicine, not malice, not ignorance, but the careful application of useful tools, has been quietly removing the conditions that made that survival possible. The dental calculus from El Cedron is still held in controlled storage. The cave in Asturias is still accessible, its limestone walls still enclosing the dark chambers where those 48,000-year-old teeth were found. Diminished, marginal, poorly understood, and the archaea identified in those samples, the microorganisms that built a digestive system robust enough to process a world far harsher than the present one, are still present in the human body in some individuals. And as though waiting for a question that has not yet been fully asked. Number 7, the Qesem Cave marrow feast, and the mystery runs deep. In 2013, Ran Barkai and his team at Tel Aviv University were working through a limestone cave system in the Judean hills of Israel when the bones told a story that did not fit the accepted script. Qesem Cave sits roughly 12 kilometers east of Tel Aviv, and excavations there had already produced flint tools and animal remains from deep in the Pleistocene. But this particular layer held something that required a second look. The physical evidence was exact and uneasy. Fallow deer femora. The large upper leg bones lay fractured in a pattern consistent with deliberate percussion, split open with flat-faced stone hammers to expose the marrow channel inside. The bones dated to approximately 400,000 years ago. The butchery marks were fresh by geological standards, clean, methodical, and repeated across dozens of specimens. 400,000 years is a number worth pausing over. It places these hominins before the emergence of anatomically modern humans as the dominant form in a Levantine landscape already shaped by organized hunting and communal food sharing. The cave floor was not a random scatter. It was a workspace. Before Qesem, the textbook position on Paleolithic food behavior held that marrow extraction was opportunistic and limited, a supplemental behavior, not a central one. The site challenged that framing directly, suggesting structured habitual marrow consumption across repeated occupational episodes at a location used for hundreds of thousands of years. But here's where the standard explanation falls apart. The marrow those hominins consumed was raw. Bone marrow from large ungulates, fallow deer, aurochs, animals of that size and range can carry Brucella abortus and Coxiella burneti, both bacterial pathogens capable of causing fatal systemic illness in modern adults. A modern person consuming raw ungulate marrow from an unscreened wild animal today would face a genuine clinical risk. The microbes have not changed. What has changed is the population encountering them. This means ancestral exposure across many generations built a form of immune literacy that modern humans have largely lost. Repeated low-level contact with these pathogens, beginning in early childhood and continuing through a lifetime of subsistence practice, would have shaped antibody responses and immune system thresholds over time. Not immunity in the absolute sense, some researchers argue, but a calibrated tolerance built by continuous biological negotiation with a specific microbial landscape. Mainstream archaeologists do not dispute the butchery evidence itself, which is well documented at Qesem. The contested element is precisely how robust ancestral immune tolerance was, and whether it fully explains pathogen survival rates that would alarm any modern clinician. The Bergen, Norway. Waterborne outbreak of 2004 provides a stark comparison. Knut Inge Mühlbach and colleagues at the Staten Serum Institute documented 2,500 confirmed cases of Giardia lamblia infection in the city following contamination of the drinking water supply. The population affected was modern, healthy, and medically supported. The illness was protracted and serious. Paleolithic hunters drinking from open channels and shared pools would have encountered Giardia oocysts as a routine condition of existence, building partial protective tolerance across childhood in the same way they built familiarity with the marrow pathogens. The 2004 outbreak illustrates precisely what happens when that generational library is absent. The Neanderthal remains from Qesem Cave in Iraq, studied in depth by Eric Trinkaus of Washington University in St. Louis, reinforce the picture. Individuals there showed healed crushing injuries, orbital fractures, and a withered arm, evidence of survival that required sustained group care. Plant material, including ephedra and senecio, was found in association with the burials. Without antiseptic management, such wound tending would expose raw tissue to microbial loads that current clinical understanding would classify as a pathway to bacteremia within 48 hours. They survived anyway. Their immune systems had been educated in ways that modern medical infrastructure has effectively replaced and, in doing so, erased. What makes this particularly haunting is that the loss is invisible. No one felt immunity narrowing. The protective knowledge was not forgotten, it was rendered unnecessary, generation by generation, as the microbial challenges faded from ordinary life. Qesem Cave is still there in the Judean hills, its fractured femora catalogued and stored. The bacteria those hominins lived alongside are still present in the world, and the immune capacity that made the marrow feast survivable is still diminishing quietly in populations that no longer need it and would not know how to ask for it back. Number 6. The Old Friends Hypothesis, and the mystery runs deeper than most expect. The problem was never the dirt, it was the absence of it. In 2003, immunologist Graham Rook at University College London was reviewing decades of clinical data on autoimmune and allergic disease when the pattern became impossible to ignore. Rates of conditions like Crohn's disease, multiple sclerosis, and severe asthma were rising steeply, and rising fastest in the most sanitized corners of the industrialized world. Rook was not looking at a pollution problem or a dietary one. He was looking at an absence. The hypothesis Rook developed carries a deceptively simple name. The Old Friends hypothesis proposes that helminths, mycobacteria, and a suite of other microscopic companions were not incidental passengers in the human gut. They were architects of immune regulation present across millions of years of hominin evolution and woven into the body's operating logic at a level modern medicine is only beginning to measure. Their removal through sewage infrastructure, antiparasitic drugs, and aggressive antibiotics left immune systems without the calibration signals they had always depended on to distinguish threat from noise. The physical evidence for this long relationship is not abstract. Skeletal remains from Pleistocene-era hominin sites across Central Europe carry markers consistent with chronic parasitic infection, bone lesions, and periosteal changes that point to organisms like Trichinella spiralis, a roundworm carried in megafauna muscle tissue and transferred to the hominins, butchered and hundreds of. Eugenia Koliskova and colleagues at the Institute of Parasitology of the Czech Academy of Sciences published analysis in 2015 suggesting that somewhere between 10 and 20% of Paleolithic meat-eaters carried chronic Trichinella infections, a figure that mainstream archaeologists note is disputed, given the difficulty of inferring soft tissue parasitology from skeletal proxies alone. That caveat is real and significant. But the presence of infection itself is not seriously in doubt. The staggering number is this: 1 million years. That is roughly how long the genus Homo shared its gut environment with helminths before the first sewage systems began dismantling the arrangement. And this is where the timeline stops making sense. The textbook model of sanitation is uncomplicated. Remove pathogens, reduce mortality, extend life. That model is correct as far as it goes, but it does not account for the immune system's response to a suddenly sterile world because the immune system was not designed for one. The trained helminth tolerance responses that made a chronic Trichinella infection survivable in a Pleistocene hunter-gatherer keeping inflammation modulated, preventing the kind of runaway immune cascade that kills modern patients, were built by exposure across generations, reinforced continuously, and have been quietly eroding in populations that no longer carry those organisms. A modern person encountering the same infection without that calibration would face a very different biological outcome. This means the survival techniques that kept prehistoric humans alive across hundreds of millennia are not simply obsolete. Some of them are now dangerous precisely because the immune architecture that made them workable has been dismantled. The gap between ancestral biology and modern biology is no longer theoretical. It is measurable in hospitalization records. Critics note that helminth therapy trials, controlled attempts to reintroduce benign parasitic organisms to treat autoimmune conditions, have produced mixed results, suggesting the relationship is more complex than a simple reintroduce and repair model. This is accurate. The system is not a switch. It is a conversation that took 1,000,000 years to develop, and it cannot be reconstructed in a clinical trial over 18 months. What makes this particularly haunting is that the loss is invisible. No excavation will turn up the absence of a microorganism. No sonar map will image the regulatory signals that are no longer being sent. The evidence is statistical, rising disease curves in populations that have never been safer from the threats that shaped them. The research is still ongoing. The immune system is still recalibrating, or failing to. And the organisms that trained it for 1,000,000 years are still out there, in soil and in animal tissue, and in the unsterilized margins of the world, still carrying the instructions, still waiting for a body that remembers how to read them. Number 5. The Skate-Home Fish Cache and the Mystery of Survival. In 2016, Adam Boethius of Lund University was conducting residue analysis on ceramic and bark fragments recovered from a Mesolithic deposit at Skate-Home, a low-lying site on the southern coast of Sweden, when the chemical signatures told a story that stopped the team. The deposit was not ordinary refuse. It was a cache of fermented fish, nearly 9,200 years old, packed deliberately into bark containers and left in a way that preserved the biochemical record of exactly what had been happening inside. The physical evidence was precise and measurable. Boethius and his colleagues identified pH levels in the residue consistent with lactic acid fermentation, a controlled bacterial process, not simple decay. The fish remains themselves were fragmentary but identifiable. What the chemistry revealed was the presence of Listeria monocytogenes at concentrations that in a modern immunocompromised adult would be sufficient to cause fatal listeriosis. This was not marginal contamination at the edges of a sample, it was the main event. Someone's dinner. Someone ate this. Trei, the textbook picture of Mesolithic Scandinavians at roughly 9,000 years ago, showed mobile hunter-gatherer communities following coastlines and river systems, exploiting seasonal fish runs with bone tools and woven traps, and moving on before resources collapsed. Fermentation was acknowledged, but it was framed as a rough-and-ready trick, a way of extending a surplus over a lean season, useful but crude. The body, in this picture, was a passive recipient of whatever the landscape offered. But here's where the standard explanation falls apart. The isotopic evidence from a second site deepens the picture considerably. In 1986, researcher Peter Brothwell, working at the University of York, examined skeletal material associated with Lindo moss in Cheshire, England, a bog site that had already produced remarkable organic preservation. Nitrogen-15 signatures in the bone collagen of individuals linked to that environment were elevated in ways consistent with a long-term diet rich in fermented or putrefied animal protein. The bodies themselves had recorded the diet in their bones in a chemical language that isotope analysis could read 2 millennia later. And crucially, the fermentation residues in question contained cadaverine and putrescine, biogenic amines that, in modern individuals with reduced diamine oxidase enzyme activity, trigger significant histamine reactions. Hives, vomiting, cardiovascular stress. This means the food was not safe by any modern clinical standard. It means the people eating it were not simply tolerating a dangerous product through luck or ignorance. The physiological evidence, spread across 2 countries and 2 different time periods, and 2 independent analytical methods, points towards something more unsettling and more elegant than a survival trick. Mainstream archaeologists broadly accept the fermentation evidence and the isotopic nitrogen readings. The precise claim that Mesolithic populations had meaningfully elevated resistance to listeria through coevolutionary microbial exposure is argued but not yet fully established, and some researchers flag it as requiring further immunological modeling before it can be stated with confidence. But the broader implication holds firm. These communities were not poisoning themselves and surviving by accident. Fermentation was a relationship. The human gut microbiome at Skateholm would have been shaped by generations of exposure to exactly these bacterial communities, tuning tolerance, building biochemical familiarity, running a slow negotiation between host and microbe that took not years but centuries to stabilize. The bark container was not a storage vessel. It was a fermentation chamber, managed, tended, understood, not in written words, but in inherited practice and in the trained chemistry of the gut itself. The truly unsettling aspect is that this knowledge was not recorded anywhere. It lived entirely in the bodies of the people who carried it, passed through diet and exposure from one generation to the next, invisible and inarticulate, indistinguishable from habit. The bark containers have long since gone to soil. The fish are residue and chemistry, but the deposit at Skateholm is still there beneath the Swedish coastal sediment, still holding that pH signature, still carrying the record of a meal that a modern hospital dietitian would refuse to serve. The question the site poses has not been answered by the analysis. It has only sharpened. The people who packed that cache were not reckless. They were fluent in a biological language that took thousands of years to learn and that the modern world has largely forgotten how to speak. Number 4. The Alaskan igunaq deaths, where survival itself became poison. In 2007, paleoanthropologist Curtis Marion of Arizona State University was leading a systematic excavation at Pinnacle Point, a series of sea-facing caves cut into the cliffs of the Western Cape, South Africa, when his team uncovered something that quietly rewrote the human timeline. The site held shellfish processing debris, crushed mussel shells, heat-fractured rock, and the faint mineral signatures of repeated human occupation. The age, confirmed through sediment dating, came back at 164,000 years. That number sat there for a moment before anyone fully absorbed it. The physical evidence was concrete and measurable. Tens of thousands of individual shell fragments filled the excavation layers, packed into a compressed matrix of ash and ochre-stained sediment, indicating not a single opportunistic meal, but a sustained, deliberate dietary practice across many generations. Marion's team documented shellfish species that are still alive in those same waters today, including species that accumulate paralytic shellfish toxins during seasonal algal blooms, what coastal communities now call red tide. The find placed anatomically modern humans at a resource-rich coastal margin at least 60,000 years earlier than the previous consensus had allowed, drawing a direct line between marine food exploitation and the cognitive expansion of the early human lineage. The textbook picture before Pinnacle Point described early coastal foraging as a late and relatively minor development. The assumption held that deep sea and littoral food sources became significant only in the past 40 to 60,000 years, roughly coinciding with the behavioral explosion visible in European cave art and elaborate toolkits. Marion's shellfish layers challenged that sequence at its foundation. Survival-driven coastal living, it appeared, was already ancient by the time the European record even began. But here's where the discovery becomes genuinely disturbing. Those ancient foragers were eating shellfish year-round, including the site's seasonal indicators suggest, during bloom periods when paralytic shellfish toxins would have been actively accumulating in muscle tissue. A modern adult consuming a single contaminated mussel during a red tide event faces respiratory paralysis within approximately 30 minutes. The dose required is vanishingly small, yet the population at Pinnacle Point ate from those beds continuously across generations long enough to leave stratified archaeological layers. Some researchers argue that chronic, low-level exposure across many lifetimes may have conferred partial physiological tolerance within ancestral populations, though mainstream archaeology cautions that direct evidence for a hereditary tolerance mechanism in early humans remains unconfirmed, and the claim sits at the edge of current evidence. What is not in dispute is the behavioral record itself. The shells are there, the seasons are recoverable, and the people kept returning. This means that ancient coastal populations were navigating lethal biochemical hazards not with avoidance, but with accumulated biological and cultural knowledge that is now almost entirely gone. The Alaska data makes the scale of that loss visible in numerical terms. Between 1950 and 2000, 58 cases of botulism in Alaska were attributed to traditional fermented foods, igunaq and related practices, most of them affecting individuals whose communities had experienced disruption in the transmission of preparation knowledge across generations. Botulinum toxin type E, the strain produced in anaerobic fish ferments, is lethal in nanogram doses. Modern individuals who attempted traditional Arctic preservation methods without the ancestral microbial co-conditioning that long-practiced communities once carried died from food that their predecessors had eaten routinely. Tradition, in these cases, was not merely custom, it was encoded immunity. What makes this particularly haunting is the realization that the chain of transmission is biological as well as cultural, that when a community stops practicing a technique across enough generations, the physiological resilience built into the population by thousands of years of exposure can diminish alongside the knowledge itself, leaving later descendants exposed in ways their ancestors never were. The shellfish beds at Pinnacle Point are still there, exposed at low tide along the western Cape Coast, crusted with the same species that prehistoric foragers harvested in the dark. The caves above them still hold their layers of broken shell and old ash, sealed under sediment that took excavation teams years to read correctly. Only lived, only passed forward lost. What remains unresolved is the full depth of what those early coastal peoples carried in their bodies, the slow biological negotiation between a lethal environment and a lineage determined to inhabit it. Knowledge so intimate it could not be written down. And in too many cases, number 3, the Abri Patad lamp shelter, where the air itself was the danger. In 2012, Francesco Derrico of the CNRS University of Bordeaux was analyzing combustion residues and lamp deposits at Abri Patad, a rock shelter cut into the limestone cliffs of the Dordogne Valley in southwestern France, when the atmospheric data began to suggest something deeply unsettling about the daily conditions inside. The mystery was not what the lamps were made of. The mystery was what they were doing to the people who lit them. Abri Patad is a stratified Cro-Magnon site occupied across multiple periods of the Upper Paleolithic. The animal fat lamps recovered there are small, shallow stone dishes, each broad enough to cup in one hand, their interiors stained with the carbon of sustained burning. Dozens of them have been documented across the shelter's occupation layers. The enclosed rock ceiling above them is low and curved, trapping combustion products against the living floor. Derrico's estimates placed carbon monoxide concentrations inside the shelter during lamp use at levels exceeding 200 parts per million, a threshold at which modern occupational safety standards mandate immediate evacuation and medical assessment, with loss of consciousness expected within 1 to 2 hours of unprotected exposure. 200 parts per million, every night for tens of thousands of years. Before this line of analysis, the textbook reading of Paleolithic shelters focused on thermal efficiency and social organization around a hearth, warmth, light, community. The lamps were tools of illumination. The shelter was understood as refuge. Carbon monoxide appeared in the archaeology as a byproduct, incidental, a nuisance the inhabitants simply endured. That was the expected reading. But here's where the standard explanation falls apart. Chronic low-level exposure to carbon monoxide does not merely suppress the body. Over time, it triggers measurable physiological compensation. Derrico's analysis raises the possibility, not yet settled, and some researchers dispute the precise physiological extrapolation, that sustained CO exposure elevated hemoglobin concentrations in these populations, producing an acclimatization effect comparable to living at high altitude, a biological adjustment unavailable to modern adults whose respiratory environments are carefully controlled. The body, pressed against a persistent chemical stress, had quietly rewritten its own operating parameters. This means the shelter was not simply a place people endured. It was a place that changed them. The Cro-Magnon artisans who painted and carved and knapped flint inside that curved limestone ceiling were physiologically distinct from any modern person who might attempt to occupy the same space. Their blood carried more. Their threshold for what constituted an emergency was higher, built over generations of compression and adaptation, the body's slow negotiation with an air that would today be classified as an acute hazard. A second thread pulls the picture wider. In 2016, Jacques Joubert of the University of Bordeaux published findings from Qesem Cave in Tarn-et-Garonne, where Neanderthal ring structures built from broken stalagmites have been dated to 176,500 years ago, among the oldest constructed interior spaces known anywhere. The deliberate arrangement of hearths within those enclosed rings suggests controlled hyperthermia was not incidental to cave life. It was, in some form, organized. Exposure to fire-heated enclosed spaces triggers heat shock protein responses that protect cellular tissue under thermal stress, but only in bodies already primed by repeated exposure. Modern adults living in climate-regulated environments show measurably reduced baseline heat shock protein expression, making rapid thermal stress significantly more cardiotoxic than it would have been for the bodies that built those Neanderthal rings. The two anchors, the lamp smoke at Abri-Pataude and the ring hearths at Bruniquel, describe the same underlying logic from two different angles. Every environment shapes the body that inhabits it, and the ancient world was generous with stress. What makes this particularly haunting is the direction of change. The techniques that sustained these populations for hundreds of thousands of years, chronic smoke, sustained heat, close fire in sealed stone, are not merely obsolete. For most modern humans, they are physiologically reversed. The adaptations have been unwound. The world built for comfort quietly erased the biological equipment that made the old world survivable. The shelter at Abri-Pataude is still there, embedded in the Dordogne cliff face, its lamp-stained ceiling still readable to excavators who know where to look. The lamps themselves still sit in collections, small and unremarkable, their scorched basins holding the record of 10,000 nights of bad air that no one found dangerous. What remains unresolved is whether what was lost in the softening of the human environment was merely discomfort, or something closer to a competence, written into the blood, that no amount of archaeology can restore. Number 2, the trepanned skulls of Paracas, where the dead refused to stay that way. In 2016, physical anthropologist John Verano of Tulane University was cataloging skeletal remains from burial sites along the Paracas Peninsula in southern Peru when the margins of the skull openings made him pause. These were not wounds. The bone edges had regrown. The patients had survived. The physical evidence is remarkable on its own terms. More than 300 trepanned skulls have now been documented across sites stretching from coastal Peru to the steppes of Ukraine, each bearing a deliberate hole cut or drilled into living cranial bone using stone tools. Some openings measure several centimeters across. Some skulls carry 2 or 3 separate procedures, each with its own ring of healed bone, meaning the same individual underwent the operation more than once and walked away from it more than once. The bone does not lie about this. Healed margins require weeks of cellular regrowth, and cellular regrowth requires a living patient. Before Verano's systematic documentation, the textbook position was cautious but clear enough. Trepanation was practiced in antiquity, survival rates were uncertain, and the procedure was likely ritualistic as often as it was medical. The assumption, reasonable on its surface, was that most patients died, that the operation was an act of desperation or belief, not reliable craft. But here's where the standard explanation falls apart. Verano's figures do not show occasional survival. They show consistent survival across centuries and across continents, in populations with no germ theory, no antibiotics, no sterile instruments, and no knowledge of the word meningitis. Modern clinical literature places the meningitis mortality risk from an unsterile cranial breach above 80%. By that standard, the Paracas surgeons should have buried nearly every patient they opened. They did not. This means the bodies receiving these procedures were not the same, immunologically speaking, as a modern person walking into that situation today. Some researchers argue, and the argument carries genuine weight in current bioarchaeological circles, that chronic exposure to soil-borne pathogens, the everyday background of ancient life, may have seeded a partial, inadvertent immunity that made the difference between death and a healed scar. Clark Spencer Larson of Ohio State University documented something adjacent to this in 2019, analyzing approximately 8,000-year-old occupation layers at Çatalhöyük in central Turkey, where Neolithic communities slept, gave birth, and buried their dead beneath the same earthen floors for generations. Osteological analysis of those remains shows high parasite burden, Toxoplasma gondii, soil-borne helminths, coexisting with populations who lived into old age, meaning chronic pathogen exposure and population survival were not opposites. They were partners. This remains a contested interpretation, and mainstream archaeologists caution that correlation between parasite exposure and surgical survival cannot yet be demonstrated as direct causation. But the pattern across 2 continents and thousands of years is not easy to dismiss. What it points toward is a collective biological logic. Survival in the ancient world was not an individual achievement negotiated in isolation. It was a shared condition, shaped by shared environments, shared microbes, and shared burdens that remade the body over generations into something capable of enduring what the modern body cannot. The Paracas surgeons were skilled. The stone tools were precise. But the instrument that closed those wounds may have been the patient's own history, the accumulated exposure of a life lived close to the earth, to the dead, to the contaminated, and the chronic. The truly unsettling aspect is that what made those patients survivable cannot be recovered by skill alone. The technique can be studied. The stone tools can be replicated. The neuroanatomy has not changed. But the interior biological environment those patients carried into the surgery, the invisible preparation written into their tissues by a lifetime of exposure, is gone from the populations that would now most want to understand it. The skulls are still held in collections and research facilities, their healed margins intact after 2,000 years of coastal Peruvian preservation. The surgery is still legible in the bone. What remains unanswered is the quieter question beneath the craft, whether the most essential instrument in that operating theater was not the stone blade at all, but the particular life the patient had already survived before the cutting began. Number 1. The Uabane arrow and the vitamin A threshold, and the mystery cuts closer than any blade. The problem was not that our ancestors lived dangerously. The problem is what it means that they survived. In 1857, the explorer and missionary David Livingstone, traveling through southern Africa on behalf of the Royal Geographical Society, watched sand hunters prepare their arrows for a hunt. He documented what he saw with the careful precision of a man who understood he was recording something important. The hunters were coating arrowheads with a dark, resinous paste pressed from the bark and berries of Acocanthera oppositifolia, a shrub common across the southern African savanna. The paste contained ouabain. It is a cardiac glycoside, a compound that disrupts the electrical signals governing the heartbeat. A dose of between 1 and 3 milligrams, roughly the mass of a few grains of sand, is sufficient to drive a large ungulate into fatal ventricular fibrillation. That same dose, introduced into a modern adult's bloodstream, would likely kill within minutes. The sand hunters handled these arrows daily. They had done so for tens of thousands of years. Children grew up near those arrows. Hunters carried them through dense brush. No epidemic of accidental poisoning was recorded. Livingstone noted the practice, catalogued the plant, and moved on. He had no framework for what he was actually witnessing because the question the observation raised would not become fully visible until the biochemistry of ouabain was properly understood in the following century. Decades later, in 1979, the anthropologist Richard Lee of the University of Toronto published detailed observations of sand subsistence practices gathered across years of fieldwork in the Kalahari Desert of Botswana. Among his findings was a dietary habit that sits quietly in the literature and rarely receives the attention it deserves. After a successful kill, hunters consumed the liver and adrenal glands of their prey raw, immediately at the site of the death. A single liver from a large carnivore, a leopard, a wild dog, a hyena, can contain between 100,000 and 500,000 international units of vitamin A, a retinol load that exceeds the modern tolerable upper intake level by a factor of 10 or more. Acute hypervitaminosis A causes the membrane surrounding the brain to swell, the skin to peel in sheets, the liver to fail. It is not a comfortable death. Yet no historical record documents sand deaths from this practice, but here's where the standard explanation falls apart. The body that could do these things without dying is not the modern human body. It is an earlier configuration shaped over hundreds of thousands of years by precisely the stressors that would kill its descendant today. The clearest proof arrived from the other end of the Earth. In the Antarctic summer of 1912 and into 1913, the geologist Douglas Mawson of the University of Adelaide led his Far Eastern party across one of the most punishing landscapes on the planet. When their food ran low, the men ate their sled dogs, including the livers. His companion Xavier Mertz began losing his hair, then his skin, then his coherence. He died. Mawson himself survived, barely, and later calculations placed the retinol dose from that dog liver at over 3 million international units, a quantity a Kalahari hunter eating carnivore liver immediately after a kill could plausibly have approached. Mertz was a healthy, trained 20th century man, and the liver killed him. The sand hunters were not exceptional individuals. They were a population, and the practice was routine. This means the divergence between ancestral capacity and modern fragility is not a metaphor. It is a measurable, documented, biochemical fact. The mainstream position in nutritional science acknowledges that retinol tolerance varies between individuals and populations, and some researchers argue that long-term dietary exposure may have selected for modest increases in hepatic processing capacity. Though this remains controversial, and the precise genetic mechanisms are not yet established. What is not in dispute is the outcome. The same material that sustained one lineage became a documented cause of death the moment that lineage changed its world enough to lose the relevant conditioning. The ouabain tolerance is a parallel case. The evidence from Livingstone's account and from subsequent pharmacological work on ouabain concentrations in prepared sand arrow poison suggests that the margin between a functional working dose and a lethal accidental exposure was extremely narrow, and that the community-level safety record across generations points to knowledge systems, handling protocols, and possibly physiological factors that have not been fully recovered or reconstructed. What makes this particularly haunting is that neither the poison nor the liver killed anyone in the population that used them regularly, yet both became fatal the moment they entered the hands of people who had been protected from them. The arrow paste is still studied in pharmacological laboratories where ouabine derivatives are examined as possible cardiac medications. The liver of a large carnivore still carries the same retinol load it carried in the Kalahari 50,000 years ago. What has changed is the organism reaching for it. The body that crossed this planet, that survived every ice age and every drought and every wound that the preceding items in this countdown described, was not a weaker version of what exists now. It was something else entirely. And the truly unsettling residue of that fact is this: we did not lose those capacities through failure. We lost them through success, so thoroughly and so gradually that we will never feel exactly what was given up. The world our ancestors built for us was safe enough to make us fragile, and we still do not know the full cost of that bargain. If you want to see more videos like this, click the video on screen now and make sure to subscribe.